Yes, thank you.
Sorry. সুনেত্রা দি,
হ্যাঁ, বলো।
বলছি আমরা কি এখানে এই উইন্ডোতে, গুগল
হুম।
মিটের এই উইন্ডোতে ওই ইয়েটা দেখতে পাচ্ছি না, যে প্রেজেন্টেশনটা আছে।
না, ওটা এখনো আমি দিয়েছিলাম, তারপরে এই প্রথমবার শেয়ারিং এর জন্য ওটাকে একটাকে না থামালে অন্যটা হচ্ছিল না, তাই এবার শেয়ার করে দিচ্ছি।
ওকে, ওকে, ওকে।
হ্যাঁ, ও তো বেরিয়ে গেছে, ও
আমি,
জয়েন করতে বলো ওকে। এমনি এ করে,
হ্যাঁ,
মানে এসে
ওকে।
না শেয়ার করলেই হলো। হুম।
হুম,
ওকে,
হুম।
আমি শেয়ার করে দিচ্ছি ওটা।
ঠিক আছে, আমি বরং একটা মেসেজ করে দিচ্ছি, যে participants shouldn't be typing anything or sharing screen.
নমস্কার, আমার প্রিয় ছাত্রীরা, সতীর্থক অধ্যাপক।
Hello? Hello? Hello?
Hello? Yeah.
Yeah, am I audible?
Yes, yes, you're audible.
Okay, okay. Um, Ma'am, hello, Principal Ma'am. Hello, Madam. Ma'am is here. Shijati, hello.
Ma'am
Yes,
is
yes, hmm,
Is Ma'am here?
yes,
Has
I'm
Ma'am
here.
joined? Hello.
Priya, take permission of principal ma'am and then you can, I think, start. Our speaker is here, so
Yeah, I
we can
I
start, I
was just
think.
thinking if Principal Ma'am is there because
Hmm. So she's calling me, just a minute.
actually.
Principal ma'am is joining. There was some problem with her audio and video. She has left and again
Okay.
joined. Hmm.
Okay, all
Now
right.
she has joined again. Ma'am, please check whether your uh,
Hello.
Just a second, some problem is there with principal ma'am's. Principal ma'am, can you hear us?
To all of you who have joined us, given your kind time to us to join in the session today, we'll be starting the session shortly. Since this is a absolutely technology based program, so we'll have to just bear by some of the glitches. Meanwhile, we're just waiting for our principal ma'am to join, and soon as she's connected, we'll start our session. Thank you.
Please stay connected with us. We are trying to resolve the technical issues. Thank you.
Hello?
Hello,
Yes. Yes, you're audible
audible?
now. Yes.
Hello.
Okay.
Hello, ma'am. Am I audible to you?
Yes, yes, you are audible and
So
audible. Okay. Then
we
we will
have
start.
we have our speaker with us. So should I start?
Yes, start.
Okay.
My goodness. Please put the audio muted.
Hello.
Hello.
Yeah. Okay, ma'am. So let's start then.
Yes, let's start.
Okay, ma'am. Hello and a very good evening to all of you present here today with us, and also to those who will be joining with us shortly. I am Dr. Priya K. Gopal of Department of Microbiology, Muralidhar Girls College. It is my utmost pleasure and honor to welcome all the participants, my respected colleagues, seniors, my dear students, and our esteemed speakers to the international webinar Convergence of Biological and Medical Sciences, a Ray of Sunshine in COVID Pandemic 2020, jointly organized by the Department of Chemistry and Microbiology. To begin our webinar today, I would like to call on our respected Principal Madam, Dr. Kinjalkini Bishash, to deliver her welcome address. Over to you, ma'am.
Yes, thank you, Priya. A very good evening to all of you. And on behalf of Muralidhar Girls College, I extend a warm welcome to all my resource persons, participants, colleagues, students to this webinar titled Convergence of Biological and Medical Sciences, a Ray of Sunshine in COVID Pandemic 2020. I would like to thank the organizers, the joint organizers of the Department of Chemistry and that of Microbiology, with special mention to my faculties, Dr. Shejuti Mondol, Dr. Priya K. Gopal, Srimati Ambalika Bhattacharya, Srimati Jhilmil Bhattacharya, Dr. Sharat Patra, and Dr. Deep Mondol for their enthusiasm and untiring effort to organize this international webinar. Also, the technological help and advice by the college ICT cell needs special mention. This is all an in-house effect, and you see we haven't taken the help of any other outside sources. Now, it is definitely a great honor to host this webinar with such eminent speakers. Dr. Shomaditya Bandyopadhyay, who is a doctor in geriatrics and is presently associated with the Royal College of Physicians, and has done his diploma from King's College London, and is a member of the clinical trial staff of the recovery trial. He himself is a COVID-19 survivor, and we hope to be enriched by his first-hand news regarding this COVID situation. Also, we have with us our next speaker, Dr. Shantanu Paul, Center for Advanced Studies, Department of Botany, Calcutta University. He has also been associated with us, with this college, for the past 4 years, being a governing body member of this college and knows us. Now, with a positive note, and mind you, not the corona positive one, as you see, the mere concept of positive and negative have reversed its role in this COVID Juke. All throughout our lives, we were to believe that the positivity should be our cup of tea to live life. But now, being tested positive is a bad thing. Rather, negative test results should prevail and makes us happy. So I sometimes ponder what will be the situation after this COVID Juke. Will the world prosper in humanity or it will face a great loss? I have written it down in a short poem which I would like to share with you. Now, it goes as such. An innocent question to the Lord. Is it for the betterment, prosperity of the humanity to belong to a COVID youth with no new clothes to fill your closet, no new ornaments to deck you up, no extravagant, luscious parties, dinner, invitations, get-togethers, public appearances? Not at all necessary. Spending millions unnecessarily. When the other side endures extreme poverty, so much so that having a single meal a day, that too insufficient, with no stable roof over your head, in threadbare clothes, striving to life, living in its meager terms. When one embarks on simple living with simple healthy food, healthy ways, doing the course of your own household lovingly, enthusiastically, and with quote unquote time, the time which every family member craves from you, meditating, concentrating on you, indirectly to one's Atma, a certain peace prevailing. A question to you, my Lord. So I would like to thank all for our patient cheering, and I wish that this seminar will reach its heights. It has received an overwhelming response from such a short period of time, and I hope that this success craves for more along with these lines. And I would like to hand it over to Priya for the next part. Over to you, Priya.
Thank you so much, madam, for your glorious and delightful welcome address. On that positive note, I'd like to take the session further from here, and I'd like to call on our first speaker of the day. Today, we are lucky to have on board with us Dr. Shantanu Paul, Professor of Botany at the University of Calcutta. His areas of specialization include cancer biology, genetics, phytochemistry, and immunology. He did his PhD from the Indian Institute of Chemical Biology, Jadavpur, Kolkata in the year 2003. Thereafter, he worked as a postdoctoral student at the prestigious Feinstein Institute of Medical Research, New York, USA, till 2008. He is presently involved in extensive research and currently supervising several major research projects sanctioned to him. He has successfully mentored 2 PhD students, and currently 6 students are enrolled under him pursuing their PhD degrees. To highlight a few of Sir's academic achievements, in the year 2000, he was the recipient of the Young Scientist Award for Biomedical Research in India, Perspectives and Prospects. He was selected as an associate member of American Society for Hematology for his outstanding contributions in the field of hematology in the year 2006. In the year 2017, he was selected as editorial board member for the Austin Leukemia Journal, USA. He has 2 international and 1 national patents to his name on detection of acute lymphoblastic leukemia. With a research experience of over 25 years and teaching experience of 20 years, he is also engaged in various academic and administrative responsibilities at both the undergraduate and postgraduate levels. Today, the topic of his talk is In Search of a Vaccine for COVID-19. On that note, I would hand over the session to Dr. Shantanu Paul. Over to you, sir.
Am I audible? Can you hear me?
Yes,
Yes,
yes, you're
yes,
audible.
you're clearly audible.
Okay.
Yes.
Thanks, Priya, for your kind introduction. So, good afternoon, everybody. First of all, I will thank the principal of Muralidhar Girls College, Dr. Kinjal Kini Biswas, whom I know for many years, and she has been engaged in many good activities, and also this is one of the very good activity in time of COVID. We are discussing about something at least positive for COVID. I will also thank the joint coordinators of this webinar, Dr. Priya K. Gopal and Dr. Shenjuti Mondol from the Department of Microbiology and Chemistry of this college, and all the faculty members, those who are engaged or in any way or the other, and all the other staffs of this college, because I know this college for at least 5-6 years. So it is a great privilege for me to give a talk over here. So what I will say is that, you see, because the mankind is now facing a very great, good struggle, because we are at the pandemic situation of a very novel virus, that is coronavirus. So at this time, if you see on board, India at present is third. USA is at the top one, and second is Brazil, and third is India as far as the new confirmed cases is concerned. Right now we have like 17,50,000 corona positive cases throughout India. But good thing is that of that there are 11,45,000 have recovered, and with a mortality rate of 2.42%. So amid this COVID, we will try, because I know that there are most of the participants are actually students. So I will try to touch the scientific flavor of the biological science in the making of the vaccine. Okay. So I will just start this, but before I start my presentation, I will just touch something. So I will first give an idea of the history and epidemiology of this disease in short. Then I will go to the structure of the virus, then how the virus actually infects our and comes into our body, and then how it proliferates from a good give infection from one person to another, and going to a little bit of unknown value, then how we can require herd immunity, and then the vaccination protocols and what are the different protocols used for vaccination, particularly in corona, and some of the regulations like clinical trials, and then just a short bit of punch, what are the latest updates of COVID vaccine. I know there is a doctor next to me who will be much touching on that note, so I will give a small idea of that and then I will end. Okay, so let me present this slide first. So can you see this slide? It's working?
No, sir, I not yet.
No, yes, it's visible. Visible.
Yes, now it's
Yeah.
visible. Yeah.
Visible. So I will start my talk in search of a vaccine for COVID-19. So let's come to the history of COVID-19. It started all about on 31st December 2019, when WHO was informed of a cluster of cases of pneumonia of unknown causes detected in Wuhan city, Hubei province of China. After that, the Chinese Center for Disease Control and Prevention (CDC) and the local CDCs investigate this program. The etiology of this illness is now attributed to a novel virus belonging to the coronavirus or COV family. On 7 January 2020, the coronavirus disease was identified as the causative virus by the Chinese authorities. On 11 February 2020, WHO announced a name for the new coronavirus, COVID-19. So this is the starting of COVID. But if you look in the past 20 years, there are very other viruses which are also SARS viruses, like SARS-CoV provoked a large-scale epidemic beginning in China and involving 2 dozens of countries with approximately 800 cases and 800 deaths. There is also a similar virus, MERS-CoV, that began in Saudi Arabia and has approximately 2,500 cases and 800 deaths and still causes sporadic cases. So these are in the last 20 years. So initially, the new virus was named 2019 nCoV, Subsequently, the task of experts of the International Committee on Taxonomy of Virus, ICTV, they termed it as SARS coronavirus 2, as it is similar to the one that caused the SARS outbreak, as I mentioned earlier. The SARS-CoV have become the major pathogens of emerging respiratory disease outbreaks. But on March 11, as the number of COVID cases outside China has increased 13 times, and the number of countries involved has tripled with more than 118,000 cases in 114 countries and over 4,000 deaths, who declared the COVID-19 a pandemic. So for the students, previously it was an endemic disease restricted to China. On 11th March, they name it a pandemic. So this is the difference between endemic and pandemic. So if you go just in a nutshell, I have no time so much, but viral epidemics, you see on 2002 to 2003, we have SARS, severe acute respiratory syndrome coronavirus, SARS coronavirus, and until 2012, we have the Middle East respiratory syndrome coronavirus, MERS-CoV was first identified in Saudi Arabia, and on 2009, we got a similar virus, it is coronavirus. It has like almost 87%, 92% similarity in different cases. So that's why I was talking about SARS and MERS. So what is the structure? First, we need to know the structure before we can do anything of the virus or any pathogen. So this has a very unique structure actually. This structure has, if you see, there are different types of protein basically. There are, just wait 1 minute, yes. There are different types of protein. These are in yellow, these are the spike proteins. You can see these are spike proteins. There is an envelope protein transmitted in the membrane, and there are membrane glycoproteins. These are membrane glycoproteins which I am marking. So there are three types of membrane proteins which are very good important factors for inducing immune response. And then there is a RNA. This RNA, you see, this is the RNA. So this is RNA is a positive sense single-stranded RNA of 30 kbp. So this RNA and these spike proteins are very important for developing a vaccine. And the good thing is that the rate of mutation of this virus is comparatively low than other virus. So that's why vaccination program can be successful. Next, we wanted to know how this virus get inside our body. So those who are biology students say no, they need to have a receptor and a ligand relationship. So yes, this virus actually have a spike protein. This virus actually have a spike protein that I have already said, and it actually binds with a ACE2 receptor that is present on the human cell, lung cell. So on the human lung cell, there is ACE2 receptor. What is ACE2? ACE2 is angiotensin converting enzyme 2. You don't need to know the biochemistry at this moment, but this receptor actually binds with the docks with the spike protein of the virus, and then it gets inside our body. It's not so easy. If you see, this spike protein can be divided into again two parts, S1 and S2. If you see S1 and S2, S1 is very important. S1 is the side, those who know something about, so this is the amino terminal portion, and this is the carboxylic side of a protein. So in the amino terminal end, the S part of the spike protein contains a very important thing, receptor binding domain, RBD domain, very important for evoking antibodies. Okay, it interacts with the host cell receptor and ACE2. This is the portion that actually binds with this our host cell receptor ACE2. And what is the function of the carboxylic end of the protein? It mediates fusion between the virus and the cell membrane. So the fusion part has been done by this S2 portion of the spike protein. So spike protein is very important, and particularly the RBD domain, receptor binding domain, present in the S1 part of the spike protein. It can induce neutralizing antibody that is needed for vaccine development. So this is from the structure we can get it. So this is not the all. You need to know. So we have on our lung cell ACE2 receptor, but there is also another protein molecule that is called TMP RSS2, What is TMPRSS2? This is a transmembrane serine protease 2. It has a different function. What it does is that it actually trims these clips, some of the amino acids of this spike protein before it is ready to dock inside the ACE2 receptor. So let's see. So what it does is that for SARS-CoV, this virus, to get enter into the cell, its S protein needs to be, this S protein, that means the spike protein, needs to be trimmed, termed S protein priming, so that the viral cellular membranes can fuse. Especially S protein priming is done by these TMP-RSS2. So these TMP-RSS2 actually splice some of the is a cellular proteases and it trimmed some of the portion of the spike protein, thus made it available to bind to the ACE2 receptor, and then it comes inside our host cell in the lungs. So yes, okay, so this is the way by which the thing can virus can go inside our cell. But what is the infection potentiality? That is very important in a SARS-CoV-2 person. So how many people, one sick person is likely to infect? So any viral disease, any other disease, scientists are in search for that value. So in case of other disease, if you see, so suppose this is a red is the person which is infected with corona or any disease, viral disease, say, it can infect one, that means it is infecting one person here, or it can infect 4. So what is the contagiousness of this disease? So that is represented by the R0 value. That is called the basic reproductive ratio. What is basic reproductive ratio or R0 value? Is the average number of individuals that an affected person can give the disease to. That is, a person having corona can infect how many people? Or if infected with any other viral disease can infect to how many people? So that number is the R0 value. So it is basically a constant in a particular disease. Constant means it varies from, have a range. So suppose from 4.2 to 5.6 as a range. And R0 tells us how transmissible a virus is. So take a look of some of the viruses. One is Zika virus. As you biology students, they know they have a R0 value of 3 to 6.6. That means it can infect 3% to maximum of 6.6. That means close to 7%. Measles have a range of 11 to 18. Ebola with 2. HIV of R0 value 3.6 to 7. That means HIV can infect 1 HIV person can infect 3.6 to 3.7%. Like that way, we have chikungunya of 4.1, then other diseases and proviruses. So you can see in this category of 7 viruses, there are actually the measles, we have the highest number of R0 value, that its contagiousness is high. So if you go on a platform, so R0 value, more the R0 value increases, the contagiousness of the disease increases. That means the infection rate of the disease increases. So starting from hepatitis, it comes to Ebola with both of having 2 R0 value, then HIV, SARS, mumps, measles. So the thing is that the more the R0 value, the more is the contagiousness of this disease. Okay. Now, what is the R0 for corona? So that is the main thing for the disease we are asking. So people have reported it is around between 2 to 3. So that means if it is 2, that it can infect 2%. If it is 3, it can infect 3% and then goes on. So if basic reproductive ratio or R0 value is 0, all the individuals of a society will be healthy. But if there is one infected, particularly say this is infected with coronavirus, then that can infect at a maximum of 3. So this person can infect 3% at a time, maximum of 3, because the R0 value is in between 2 to 3. So there are many reports that gives us the R0 value. That is a statistical evaluation. So again, this 3% will go on and infect 3%. So like that way, the chain will go on. So that's why scientists are telling that we need to break the chain at the initial moment of the time. So that is what the chain break is all about. So this is how the chain will multiply 3 and 3 and 3 and it will go on. So if there is some case that those who have recovered or we have a vaccine, that means green are immune people. Green means these are immune people. These are immune people. So that can be infected but at a lower rate. So if R naught is equal to 1, at one time the disease will stop. So we now come to the herd immunity. So I'm just giving some examples. So what is herd immunity? So herd immunity occurs when a significant proportion of the population, that means we are in a population and a significant proportion of the population or the herd have been vaccinated or are immune by some other mechanism, resulting in protection for these susceptible individuals. That means that is a significant proportion of, what is the proportion of a population that needs to be vaccinated so that the disease does not spread. That means the R naught value would be 0, would be 1 or lesser than 1. That means when R naught is 1, the disease remains stable among the growth. So as our R naught value of corona is in between 2 to 3, we need to make it 1, and below 1 would be good. So that is how herd immunity can help us. Herd immunity threshold is the proportion of the population. That means we have a population and the proportion of the population that need to be immunized in order the infectious disease to become stable in the community. And it can be reached through vaccination program. That is what is all about R naught value. So how many people, so one of the things is you need to calculate how many of us in India we need to get vaccinated for this virus. So there is a calculation of herd immunity, R naught minus 1 divided by R naught, and you can multiply into 100 and that value. So if R naught, if we think that R naught is equal to 2, then we can calculate R naught minus 1 by R naught is equal to half. That means if you make it into 100, that means 50% of the population of India needs to be vaccinated. Okay, that means if we have two individual, one individual has to be vaccinated. So if the R naught value is 3, R naught value is close to 3, then we need to vaccinate 2/3 of the population. That means if there is 3%, we need to vaccinate 2%. So we need to vaccinate 50% to 60% of the population. That means if we have like 135 crores of people plus crores of people, we need to vaccinate how much? 67 crore to 88.44 crore people of India to get vaccinated in other way or the other, so that the R naught value becomes 1 or less than 1. You got my point? So that is the relevance of R naught value and the herd immunity in relation to the viral disease. So that's what in a nutshell. So when we have no immunity, the virus spreads and catches everybody by a link. But when we have some immunities, say some survivors have some serum inside their body, so they can survive. So these are so they cannot immunity. So some immunity is developing in every country, and that is towards the herd immunity when we have that proportion of the population that is vaccinated either by natural way or through a vaccination process by which the disease cannot progress forward. That is our ray of hope for future, and we are very close to that also. So for that, we need to develop vaccine. Without vaccine, we cannot go into we cannot increase the herd immunity. So what is a vaccine? A vaccine is a biological preparation that improves immunity to a particular disease. That means it contains certain agents that not only resembles a disease causing microorganisms, but it also stimulates body's immune system to recognize the foreign agents. So what the virus does is that it does not let the cell know that it is a foreign particle, and thereby the cell cannot understand. So vaccination is a process by which the cell will tell, okay, this is a foreign particle that has come, and now you need to develop something to destroy that particle. So you can do this, you can raise the immunity through, one is through natural infection. As we are moving around, we are getting some natural infection, and second part is the vaccination. So vaccination is basically, vaccination reproduces actually natural infection with fewer complications because we test before we give vaccination. So I cannot resist myself, because if I start vaccination, I have to start with Edward Jenner, the person who actually is the first person who vaccinated people with smallpox since 1796. And this is Edward Jenner injecting the gardener's son with smallpox. So you see, 1796 is this story of vaccination that started from 1796 and little bit few years ago. So 1796 and now we are 2020. So we have 220 years of vaccination history on board. But you know, this 1796, though the smallpox vaccine started, who recommends that in 1980 the smallpox was eradicated? So it took like 180 years to eradicate the smallpox. So another famous scientists and French microbiologists Louis Pasteur and Emily Ross developed the first vaccine against rabies. So these are two many pioneering people. These are the very first two pioneering people, those who have changed the world in vaccination. So if you see, these people, and starting from Jenner, all these people have developed, there are many people those who have developed a plethora of vaccines. So we have like smallpox vaccine, cholera, rabies, anthrax, typhoid, plague, diphtheria, tuberculosis, tetanus, whatnot, rubella, hepatitis B, pneumococcal conjugate, human papilloma virus. So scientists know how to develop vaccines during the last 220 years. So this method of vaccination program started way back in 1798, but people are developing technology and skills, recombinant DNA technology, modern techniques into these insights. So in short, as there are many students, I thought I can put this side. There are many types of vaccine you can develop. You can develop a whole agent vaccine. That means you can take the whole virus and try to make a vaccine out of it. That means you can kill that virus and then put it. So one of the things is that you have to put something that will not cause disease. At the same time, it will evoke an humoral response. Or you can attenuate or weaken virus. The attenuated means you keep the virus on a cell line or on its animal model, and then it goes on replicating. When it gets some mutation by which it loses the ability to give infection, so it loses the ability to infect others, then you take that virus out from there and then you put it on the human being platform on a clinical trial. So that is called the attenuated vaccine. Otherwise, we can have multivalent subunit vaccine. As people from biology, they know there are many subunits, there are many proteins which have different subunits. We can take a different subunit vaccine. We can have purified macromolecules as vaccines. We can purify a molecule like spike protein and all the other things. Toxoid, capsular polysaccharide, microbial antigens. We can also develop vaccines from DNA, from RNA, and recombinant vaccines. So these are third generation vaccines. So these are different ways of by which a vaccine can be produced. Before I go into the corner, I need to give something like some examples for the students. So if you have some inactivated or kill vaccine type, there is cholera, flu, hepatitis A, plague, rabies, and polio vaccine. These are all done through inactivation of the virus or through the kill process. If you go through the live or attenuated scheme, then we have measles, mumps, rubella, yellow fever, and polio. And now you see, polio is here also, because two different scientists actually made this vaccine. First, it was made through inactivated kill vaccine. That's why it's called Salk vaccine. It is according to the Jonas Salk. Jonas Salk was the famous scientist who developed this vaccine. Then this vaccine, which is live or attenuated, is through Albert Sabin, who developed this vaccine. This is what we now know as the pulse polio that we give to our small children, just pulsing by giving it in the mouth. So this is the our famous pulse polio vaccine. So like that way we can take exotoxins, and there are many like diphtheria, tetanus, coming out from the toxoids, capsular polysaccharides like pneumonia. Pneumonia is a very good example of capsular polysaccharides. And these are third generation vaccines with the recombinant antigen surface antigens like hepatitis B vaccine, HIV vaccine against HIV, rabies, measles. So these are different types of vaccines that we can see. So there are plethora of vaccines people know how to develop. So it's nothing new. So now come to the point of SARS coronavirus 2 vaccine. We need to expose the body to an antigen. So that is the choice of antigen. Which antigen to take that won't cause disease but will provoke an immune response that can block or kill the virus if a person becomes infected. So that is the hunt for the virus. So there are 8 types of things that are being tested over for coronavirus, starting from the virus, as I mentioned, total virus, or either inactivated or weakened. It is a viral vector, whether it is replicating within the system or non-replicating. Replicating means it goes inside your body and it starts replicating its own DNA or RNA according to the genetic material and then produce the virus of its own. And the nucleic acid vaccine, either it uses DNA or RNA or a protein base, it's a protein subunit or a virus-like particle that I will go in detail. So there are majority of the virus vaccines that are in trials today are coming from any of these 8 type of vaccine strategies. So we need to know how we develop immunity. This is a busy slide. So you just know for the students. So when a virus enters our body, we are talking about the things that touches actually the spike protein and it binds with the AC2 receptor that I described and it goes inside our human cell. Once it goes inside our human cell, it is a positive strand RNA. So this RNA comes out and it is translated to proteins, then the virus assembly takes place. After the virus assembly, then the immune response needs to be evoked. Otherwise, that virus will go on building. So how we need to develop immunity? So by involving immune response. So we have antigen presenting cells, which we call APCs. So there are 3 types of antigen presenting cell. We have the macrophages, we have the dendritic cells, we have B cells also. These are all different types of antigen present. What does these cells do? These cells actually displace some of the peptides on their cell surface through a receptor. So this is the antigen presenting cell. They are taking a portion of the virus peptide and it is displaying on the surface, and by that way it is calling the one type of helper T cells, and these helper T cells, with the help of the T cell receptor TCR, it binds and it now does 2 very important functions which is very important for vaccination. One, it has to produce neutralizing antibody, so it can boost the B cells so that it can differentiate and go for a plasma B cell and then secrete antibody that will clear the virus in one hand, and also produce some long-term memory B cells, and on the second turn it will call the cytotoxic T lymphocytes. So T cells actually are of 2 types. One is helper T cells and another one is cytotoxic T cell. So this helper cell will call the cytotoxic T lymphocyte. Why cytotoxic T lymphocyte is very important? Because these are the cells that, so antibody actually catches the antigen or the virus, but here cytotoxic T lymphocyte, they do not catch the virus or the pathogen. It actually destroys the cell that has been inhabiting that virus. That means the virus that is present within the cell needs to be cleared out, and that can be done through the cytotoxic T lymphocytes. So after this, so now I am going into the different types slowly. So as I told, so we can for coronavirus we can take a weakened virus. That means we are taking the same coronavirus, but we are passing this through different animal models. Still, it loses the capacity to do disease. Or we can do some inactivated virus through giving some treatment analysis like heat or formaldehyde. So that actually destroys the capacity to form the disease. So once inside the cell, again the antigen presenting cell will present the viral peptide, and then the immune response will boost up. So there are at least 7 teams right now working on these virus vaccines. Measles and polio are of this example, and Sinovac Biotech in Beijing, they are working in these strategies. So this is one of the strategies. So another strategy is viral vector vaccines. There are around 25 groups working on it. So what are these groups doing? Actually replicating viral vector. There are again 2 types. One is replicating, that will replicate within this host cell, and another one which will not replicate within the host cell, that will not produce the virus itself. So what is that? In this, so there are many people who are working on this strategy. So I told you the spike protein, particularly the RBD domain, what they are doing is that they're putting the gene construct over the viral genes and put it inside the host so that it replicates and produce not the same virus but only the spike protein, so that it can evoke the immune response and give boost to ours. Like that, when in a non-replicating vector also, they are actually working on the coronavirus spike protein. So like that way, there are like 25 companies working on this area of viral vector vaccines. There are also nucleic acid vaccines. 20 teams are working on this area. It is very easy. It is like a corona spike gene. You can insert a DNA or you can insert a RNA, and then you electroporose those inside the cell, and it goes inside and then develops that spike protein which can evoke an immune response. So this is 20 teams are working on DNA and RNA vaccines right now, and there are many more coming. So this is another thing, protein-based vaccines. So this is another type which I've told. So majority protein are spike protein. Most of are interested on the spike protein because those are the most abundant protein present on the surface of the virus. Then is the membrane protein. So there are like 20 teams working on this membrane protein, spike protein, and we have actually similar vaccines against SARS virus. This is already done, which protected monkeys against infection. So there are reports and there are vaccines already made before our coronavirus came into being in 2019. So this is the spike protein and M protein. There are like more than 20 teams are working right now. And this is very important, virus-like particles. So what are virus-like particles? So this is a virus, but without any genetic material, without any DNA, or in this case, without any RNA, as in case of coronavirus. So we don't have a RNA. So what it does, so it cannot cause a disease. So once inside the cell, it will evoke a humoral immune response, and then we can minimize the disease. So that's the different types of actually strategies that scientists are taking in as far as corona is concerned. So in a nutshell, for a successful vaccination, student must recognize that you need to produce a good amount of neutralizing antibodies. You need to produce a good amount of neutralizing antibodies. That means if this is a virus and this talks with the B cells and it gives so much of things, and we don't need to get into details, but at the end what we need to get B cells antibody from the plasma B cells so that it can get hold of the bacteria and phagocytose it from the body. So one of the benchmark of the vaccination protocol is how much neutralizing antibody is being prepared and how long it is being in our body. That's why there are reports that there are some cases where people are again infected with coronavirus. That means the antibody is not persisting for some of the patients. And the second is activation of the cytotoxic T cells or any T cells. So first of all, it has to activate the, as I told, helper T cells that will in turn give some good food, that is good cytokines, release some cytokines by which they can activate the cytotoxic T lymphocyte. Very important. I will tell you in detail. So after cytotoxic T cells, they have the ability to actually kill the cell that has been infected with the virus. So they crosstalk, so they display this peptide on the MHC class 1 molecule, cytotoxic T lymphocyte, and they target the cell that has been already infected with the virus. And then what they do is that the T cells, cytotoxic T cells, they release different types of perforins, granenzymes that will kill the cell. So two things you need to get into mind. One is the activation of the cytotoxic T cells and also the antibody, neutralizing antibody for a proper vaccination. So that is done, but we need to go in I will say in a nutshell about the vaccine development and testing, particularly what are the rules and regulation. So thing is that there are different stages of the development of this vaccine. First, what we are talking about right now was the laboratory and animal studies that composed of, one is the exploratory phase and two is the preclinical phase. In the exploratory stage, we will talk about whether we will take the spike antigen or the membrane protein for vaccination, or whether we will take the RNA or what, or whether we will take the virus-like particle for vaccination. Then if so, then we have to do, this can be done in some cell lines, then we can do it in some animal models. So in a preclinical animal model, we need to do this thing. So in the animal models, we can do testing, and in this preclinical model, we need to give doses and we need to have challenge studies. That means we infected the animal with this any type of thing, say spike protein, and we need to see whether antibody is coming up or T cells are coming up, and then after those are there, we need to challenge that animal with this virus and see whether it can actually block the activity of that virus. So that is called the challenge study. So these are the exploratory stage and the preclinical stage, which most of the drugs are now. So once this is done, and if we have a very good report that there is no safety concern, biases are good, safety thing is good, then IND applications, I have talked about the FDA because it's the major thing, Food and Drug Administration of USA. So they give application for development of the vaccine. So IND, in the IND application, the sponsor, that means the company, actually describe how they will make the virus, what are the laboratory reports till that they have produced, then they start the proposed study and submit a approved clinical protocol to the FDA. So once going through, if they are happy with the report, then the vaccine is subject to 3 phases of testing. That is what we are now right now in the clinical trials. So it has generally 4 phases, but first 3 is before the vaccine goes for the market. So first phase 1 clinical trial is a small group of adults with like 20 to 80 people, and this is non-blinded. That means everybody knows what they are getting, what type of drug they are getting. This is done to assess the safety of the candidate vaccine. So how much safe this candidate vaccine is. So you need to assess the thing and determine the type and extent of the immune response the vaccine provokes. And most of the drug actually fails here. Most of the vaccine actually fails here because we are thinking that the spike protein will give us the antibody, but the data may be showing no, there is no antibody. So then it will be discarded from there. If it passes this test, then a larger group of people will be infected in the phase 2 trial, and this is randomized. So control study with a placebo group. Randomized means a study design that randomly assigns participants into an experimental group or a control group. And placebo means is an inert treatment or substance that has no effect. So this is very important. Clinical phase 2 is very, very important. So those vaccination actually pass the phase 2 has a very good chance to pass the phase 3. So in this study, safety is the major issue, immunogenicity is the again issue, then efficacy, how much good this vaccine is, how much efficacy this vaccine possesses. So how much percentage of the human beings does not get infection. So then they give the details, what are the doses, what are the proposed studies, schedule of immunization, what would be the method of delivery. These are all the things that they need to tell about the clinical trials. So if again all these things are happy, then FDA will give the nod for the phase 3 trial. So every country has their own thing. So at least you have to test like 10,000 and more. And these in the phase 3 is very crucial. Randomized and double-blind study. Randomized I have already developed. Double-blind study means here the doctors, those who are giving the vaccine, do not know which type of vaccine is giving to which group of patients. And the patients also does not know what type of vaccines they are getting. So this is a double-blinded study that has been done in the vaccine for the phase 3 clinical trial. and involve the experimental vaccine being tested against a control vaccine. So it will assess the vaccine safety in a larger group of people, and it will test the efficacy of the vaccine. So efficacy of the vaccine is one thing, whether it can stop the infection, whether it can stop the virus from giving the disease. So these are the different types of thing that we say. So if all this goes well, then the FDA will, next step is, then the sponsor or the company will apply for a license, approval and licensure. After successful completion, they will submit a biologics license application to the FDA. In case of USA, I will say, and there are many regulatory boards in different countries. Then they will inspect the factory and they will see how they are doing the thing, and then they will also monitor the production of the vaccine, and then they will give license. So after this, the thing can come to the market. So once it's in the market, these are optional post licensure monitoring. So before that, after phase 3 good trial, it can come to the market. But after coming to the market, there are also different types of trials, like phase 4 trials. There, the drug company may or may not want to know whether the drug has or the vaccine has, what is the complication of the vaccine. But the FDA will know what if there is any complications. So they have a vaccine adverse event reporting system. Anybody can report, look, I have taken this vaccine, but I have this rash, and these are the different types of things I am getting. So they can report it. They can also have a vaccine safety data link. So by this way, these are the procedures by which. So in the laboratory or in the animal model, the thing is very good. But when it comes to the clinical model, FDA or any regulatory agency of that country needs to be very much careful whether they will pass it from phase 1 to 2 or phase 3. And like that way, if you see, this is actually 10 to 15 years down the road history. But for corona, scientists are working hard, simulating very hard, doing all the 220 years of expertise, putting all the scientists together with a big hub. The scientists are trying to come up with the vaccine within 1 year to 1.5 years. This is their effort. Actually, this is a run race. So where is India situated in the vaccine? So there are 155 vaccine candidates and more across the globe that are entered various stages on human trials, out of which 2 indigenous vaccines are from India. Actually, they are in the phase 1 or phase 2 clinical trials. But we in India have 30 different Indian companies who are trying to produce vaccine and fight the infection, out of which who has given approval to only 7 candidates. So just have a look on this. So you know about the Covaxin, which is a Hyderabad-based Bharat Biotech Limited, in collaboration with ICMR and National Institute of Virology, Pune, where they are using inactivated version of the virus. So this is the platform they are using. They have already gone to the phase 2 trial. And second is the Zydus Cadila Zycov-D. They are naming the vaccine as Zycov-D, Amila-based pharma giant. They are using DNA using non-replicating and non-integrating plasmid carrying gene of interest. They are in the phase 1. So these are the 2 vaccine candidates from India that have entered into the clinical trials. Apart from that, there is Indian Immunologicals Limited, Mindvax, Panacea and Hyderabad Biologicals. But one thing we are very eager to know, that is, what about the University of Oxford, that vaccine? They are with AstraZeneca, some British Swedish major pharma. But India has something. Serum Institute of India will must produce the vaccine. Okay, so these are on the start of the phase 3 trial. So this is a adenovirus vector vaccine. So this is a very good area. Maybe there is a ray of hope, and Russia is hopeful of starting. So we don't know very in details about the vaccine from Russia. They're claiming that they are almost at the phase 3 or completed phase 3, and they will start giving the vaccine. So this is adenoviral vector vaccine again. So look, this Oxford vaccine and this Russia vaccine, both are adenovirus vector vaccine. So these 2 are actually coming up very fast, and we know many records of other vaccines. But on the last, the ray of sunshine, if I talk about just a few salient features of the Jack Caddox one, that is the British vaccine, is the vaccine has no safety concerns, no unexpected symptoms, and it can produce antibody from the 14 days and peaking at the 28 days. So they can produce neutralizing antibody, and it is long term in the body, staying for 56 days. 91% of the total volunteers can neutralize the virus, so neutralizing capacity is also good, and it also evokes the T cells that I'm telling. This is the one part of the story, and second part is the evolution of the T cells and the cytotoxic T cells. So from 14 days onwards, there is the vaccine has started building, coming up with the T cells and the cytotoxic T lymphocytes. So this part they are measuring with the ELISA technique. Most of the students know, and this is done by ELISpot assay. This is ELISpot assay, by which they are actually doing what are the T cells, whether they are produced or no. So this is the ray of sunshine. So there are many drugs coming into our hand. So we are hopeful that with the 220 years of expertise the human beings have, particularly the scientists have in the area of vaccination, with the collaboration work of different type of scientists across the globe and pharmaceutical major companies like AstraZeneca, the Oxford University, the Covaxin, and many more, and Russia, we are hopeful that a very good days are coming, and we are hopeful that there would be launch of very sooner or later there would be coming, some vaccines will be coming in the market, but maybe the vaccines will pass the phase 3 clinical trial, but its mass production, particularly like a country of India with more than 134 crores of people, and you need to vaccinate 50 to 66% of the population, it will take time. But we are hopeful in the coming few months at least they will cross the phase 3 trial. And with that hope, I will end my talk, and I will thank all the audience for their patience hearing. Thank you.
Thank you, sir. So the Q&A session is open. In the interest of time, I think we can only take 2 questions. And for the rest of the questions, I'm sure students are having a lot of them. So please write to us on the email provided in the flyer, and we will have you emailed sir's mail ID where you can directly ask your questions, and you can write to him. So the first question that student has asked, sir, is what, as a student of 3rd semester, I have a query. Is R0 value important for every infective disease to know? How significant is it to know this value for planning the treatment strategies of any infective disease?
Look, R0 value is very important because R0 value gives you the potentiality of the pathogen. That means you have a disease, and that disease needs to be transferred to the healthy individual. So if your R0 value is small, then it will only, if it is below 2, then it will catch 2%. But if it's 10, then it will catch 10%, and that 1% will give another 10%. So it will go on. So the lesser the R0 value, that is definitely important. The lesser, that better. The contagiousness increases as the R0 value increases. So this is better. But to develop vaccine, that is another strategy. So you need to have something that will evoke both the humoral response, that means the B cell activation and antibody development, and as well as the cellular response, that means helper T cell and cytotoxic T lymphocyte development. So these are the 2 other aspects of the, but R0 value lesser is good for at least combating the disease in question for the governments.
Okay. Yeah, I think that. That answers, I think, her query. Another question is, sir, is the virus airborne? What is your point of view?
Look, I don't want to get into details because there are many controversy. So during this controversial area, people are talking like that. There are many reports, those who are telling, okay, it can travel in droplets. So there are many reports in droplet. So a virus can obviously grow in droplet, but how far it is going? So there are various reports from starting to 1 meter to 10 meter, depending on the publication. So there are a wide range of publications that will confuse rather. So I don't want to
Hmm.
confuse the students with how far it is going. Definitely, it can travel in the air in the droplet. Within the droplet, it can travel, but how far it's very difficult because there are many
Yeah.
reports, and also in the Nature journal, different reports. So it's very confusing.
Hmm. The last question that we can take for today is, as we have observed, the recovery rate is quite high in India compared with the other countries. Is it only the average age is 28 years in India, or there are some other reasons out there? What do you think, sir?
So I definitely think that most of the population in India of 134 is below 20. There are huge numbers. So that's one ball game. The more you're younger, the more immunity is strong. So hard immunity, if you compare India versus United States of America or any European country, England or any country, so hard immunity is much more, immunity is much more in our country because we are fighting with different types of diseases. In USA, when we are in USA, they even have a flu shot. If you have a flu, before the
Hmm.
season, they get a flu shot. And every time in our country, we used to have flu, we don't take any flu shot. So they are not exposed to this type of environment. Their environment is much more eco-friendly than us. So we are with tons of pathogen in India, particularly India and these neighboring countries. So we have the immunity. Definitely, we have the immunity. Otherwise, the mortality rate is not. We are actually at 2.42 or like that mortality rate. And I will not comment, but we don't have so much good healthcare system. We don't have economy to support. People are
Yeah.
going outside.
Yeah.
Still, we have a
Right.
2.42%.
Yeah.
So that's the herd immunity, I will say, or the immunity developed through slogging every day with millions of tons of virus and bacteria in
Hmm.
this country. That is not present in other parts of USA. We don't have this much
That's
of
right.
diseases there.
Yeah, that's right. Thank you so much, sir, for such a crisp and yet all-encompassing, uh, informative and enthralling talk. I believe this session has been as useful and insightful for all of you who are present here as much as it has been for me personally. So thank you so much, sir. Thank you
Thank you.
for giving your time. Uh, Proceeding further in the session today, I would like to call on Dr. Shenjuti Mondol of the Department of Chemistry to introduce our next speaker of the day. Over to you, Shenjuti.
Thank you, Priyadi. A very good afternoon, everyone. We may now request our another distinguished speaker, Dr. Shomaditya Bandyopadhyay, to deliver his lecture. Dr. Shomaditya Bandyopadhyay is a specialty doctor in geriatrics at Surrey and Sussex Healthcare NHS Trust. His special interests are orthogeriatrics and Parkinson's disease. Born at Barhampur, Murshidabad, Dr. Bandyopadhyay passed the Madhyamik examination from Scottish Church Collegiate School, ranking 7th in the country. After secondary from Scottish Church College, he admitted to the Medical College. Few years after his graduation, he joined Ramakrishna Mission Seva Pratishthan and became a registrar. In 1998, he went to the UK.
Hello. There has
Sheju-
been some technical issues, I think.
Shejuti, I think
Please
you are.
stay with us.
Hello, am I
Shejuti, please
audible?
unmute your.
Yes, yes, she's back. She's back. Shejuti, you're back. Huh. Shejuti, you're not audible. You have to unmute yourself. Shejuti, you have to
Hello,
unmute
am I
yourself.
audible?
Now, yes.
Hello, am I audible?
Yes, you're audible now.
Very good afternoon, everyone. We may, but audio is. Yes, yes, I have already unmuted myself. Hello.
Shejuti, you're audible. You carry on.
Okay, okay. Very good afternoon, everyone. We may now request our another distinguished speaker, Dr. Shomaditya Bandyopadhyay, to deliver his lecture. Dr. Shomaditya Bandyopadhyay is a specialty doctor in geriatrics at Sase and Sarex Healthcare NHS Trust. His special interests are orthogeriatrics and Parkinson disease. Born at Barhampur, Murshidabad, Dr. Bandyopadhyay passed the Madhyamik examination from Scottish Church Collegiate School, ranking 17th in the merit list. After passed higher secondary from Scottish Church College, he took admission to the medical college. Few years after his graduation, he joined Ramakrishna Mission Seva Pratishthan and became a registrar. In 1998, he went to the UK for higher study and training. He is now an associate member of the Royal College of Physicians. He has also done a diploma in geriatric medicine from RCT and MSc in gerontology from King's College London. He is also a member of British Geriatric Society and British Medical Association. He is one of the clinical trial staff for recovery trial. Dr. Bandyopadhyay is a keen reader of various subjects, especially history. Photography and stamp collection are his hobbies. He lives in Crawley, UK. Now it's over to you, sir, Dr. Shomaditya Bandyopadhyay. Dr. Shomaditya Bandyopadhyay.
Thank you very much for your kind introduction. Can you hear me? Yes.
Yes, sir.
Can you hear me?
Yes, sir,
Hello?
you're audible.
Are you able to listen to me? Okay, that's great. Very much.
Yes, sir.
Yes,
You're
Many thanks
audible.
for your kind introduction. And আমাকে আমন্ত্রণ করার জন্য sorry. Okay, yeah. আমাকে আমন্ত্রণ জানাবার জন্য প্রথমেই সবিনয় ধন্যবাদ জানাই প্রিন্সিপাল ডক্টর বিশ্বাসকে। আর ডক্টর সেজুতি মণ্ডলকেও ধন্যবাদ। উনি আমার স্ত্রী সুজাতার সঙ্গে যোগাযোগ করে ওনারা দুজনে মিলে আমার দিক থেকে সমস্ত ব্যবস্থা সামলেছেন। প্রফেসর পালের ভাষণ খুবই মন দিয়ে শুনলাম, খুবই ভালো লাগলো। আর অবশ্যই অসংখ্য ধন্যবাদ আপনাদেরকে যারা অংশ নিচ্ছেন এবং শুনছেন। আশা করি আমার বক্তব্য আপনাদের ভালো লাগবে। প্রথমে বলে রাখি, I'm not a specialist in COVID-19. I'm not a virologist or an epidemiologist. I'm a geriatrician. তার মানে হচ্ছে I look after people who are older. It's because older people have different kind of diseases. The same disease, when it presents in younger person, is different from when it presents in an older person, because the cells change. The physiology changes, the body changes. So that's my specialty, and I've seen some COVID patients and have treated some of them. And I work in what we call National Health Service in United Kingdom. It's a service which is free at the point of care. And what I'm going to do is I'm going to go through my presentation, but if you have a burning question during the presentation, please do not hesitate to stop me on my slides. Right. Just checking whether it is available or not. Sorry it took such a long time. So COVID-19 is the first challenge that modern evidence-based medicine has faced, where there is no substantial evidence to go on with. People are dying while the knowledge base is being developed, which gives rise to desperation amongst doctors, and also anxiety and panic amongst people. In today's webinar, we shall try to discuss some evidence-based we will have some evidence-based discussion, and towards the end, I shall discuss some common sense advice and general advice. So I'll begin with a few words on the tests for COVID-19 and then on the medications which work and especially when to use those. Then I'll spend a little time on different hypotheses based on work that is going on about medications and also including vaccines as far as I know. I'm very grateful for Dr. Paul's presentation, and I'll stick to the medical bits about the vaccines. I'll briefly discuss the difference in treatment approach in United Kingdom and in India, and then I'll move on to the do's and the don'ts in time of COVID-19, and also a little bit on home-based self-management. And finally, we'll have a chat about how we prepare for the winter. So the tests for COVID, and there are many kinds of tests available. There are swab tests which go for PCR. There are antibody tests, which are basically blood tests to check whether you have antibody or not. There are home kits, which are of two kinds. It could be with swabs, again for PCR, and antibody against again a blood test. And there are antigen tests as well, which again uses the swab. But my message is that these tests are to rule in the infection, not to rule out. In other words, if you have a test and it says that you are COVID negative, but you have significant symptoms of COVID, what we have been doing here in UK is we have been repeating the test. So if you have a positive test, it's very good. If you have a negative test, just be a bit cautious. You may still have COVID because there is a significant false negative test in all these tests in the real world. The manufacturer might claim that it is very sensitive, but in real world, we have found there's significant difference between what the manufacturer claim and what we get. And the other thing is that there are plenty of tests available, and how do you know which one works? And this is a link I have given, which is a non-profit organization based in Geneva, and they have a lot of resources on COVID-19. And one of the resources is that they have discussed the tests that are available for COVID and whether they are good or bad. If you are interested, you can have a look. So medications. One of the problems with COVID-19 is that it's a completely new disease. So it doesn't have any new medication. And if we want to develop a medication specific for this condition, it will take months, years. So what we're trying to look into the existing medications that we have and to find out whether some of those medications would be useful in treating this condition or not. So one of the medications that I would say does work in COVID-19 infection, especially in people who have severe COVID-19 and are on ventilator, is dexamethasone. Dexamethasone has been shown to lower mortality by 8 to 26% amongst those people who have been given dexamethasone in comparison with the control group. And possibly it modifies a maturing immune response, which is often referred to as cytokine storm syndrome. There is this medication called remdesivir. It is originally developed for Ebola, and it is useful in people with COVID-19 pneumonia who require supplemental oxygen and time to recovery from 15 to 11 days when compared with placebo. This effect is not seen in most. It's a waste of money. Favipiravir is originally developed for Japanese flu, RNA polymerase. Now, there are a few things. I don't know whether it is trustworthy or not. In India have done some controlled studies, and they claim that there is a significant improvement from it. At least Beacon Pharma claims that there is 96% recovery rate after 10 days in serious cases. A point that I would mention here that these trials, unlike recovery trial, are sponsored by the pharmaceutical companies, and one might argue that they have some vested interest in saying that this medication work. So just be a bit cautious about it. Convalescent plasma. I saw in the YouTube channel someone was asking about convalescent plasma. As far as the British research about convalescent plasma is concerned, this is still ongoing. It is part of at least an arm of the recovery trial. Now, I have no personal experience with this because it is mostly done by the hematologists, but the result is not out yet. There are some preliminary data from USA which shows that the treatment is safe and well tolerated. But is it effective? They are using words like, it looks promising, we are still analyzing, but I would say that if it was really working, they probably would have been calling press conferences by this time. In any case, convalescent plasma should be used in a person with COVID-19 pneumonia and possibly the cytokine storm syndrome. So it's only in very severe cases. And none of these treatments are to be used without a doctor's advice. There is some evidence what doesn't work. And one that doesn't work is hydroxychloroquine or chloroquine. It neither works as treatment and it doesn't work as a prevention as well. There is some evidence that it doesn't work as prevention. The ongoing British trial might not have any result until end of this year. The problem with hydroxychloroquine is that there are many people who are trying to promote it. And we saw this video very recently, and Donald Trump Jr. Copied it or quoted it in Twitter and had his Twitter account blocked for some 12 hours. I don't know. Someone said that these people, they are actors. I don't know whether that's true. Someone said that Dr. Stella Emanuel, the doctor speaking in this video, has previously said that the governments are run by reptiles. I cannot say whether that's true either. And I do wonder if it is correct whether Junior Donald Trump agrees with that as well. And also there are these articles in the journal. This one has come up within the last 10 days, I think, in International Journal of Infectious Disease. It is an American study sponsored by Ford Healthcare Organization, and they are claiming the same thing that the previous video was claiming. They said that treatment with hydroxychloroquine alone or in combination with azithromycin was associated with reduction in COVID-19 associated mortality. But if you go through the paper, you find that it is not a randomized controlled trial. They looked at the patient retrospectively. The hospital admission for these patients were 2 to 6 days. It's never mentioned that whether it was mild symptomatic disease or whether it is severe disease. So the question is that how can they claim that their result is better than the fact that the natural history of the disease shows that 80% of the affected people has mild symptoms and may not need any treatment at all. And here we have the evidence that hydroxychloroquine doesn't work. This paper is from June, beginning of June, and they did it in a very important way. They got 821 volunteers who were exposed to someone with COVID-19, and they gathered these volunteers through social network. They randomized those patients to a hydroxychloroquine arm and a placebo arm. And 107 of those volunteers developed symptoms which are consistent with COVID-19. But surprisingly, the PCR swab test was positive in less than 3% of the total number of participants. And they found that there was no statistically significant difference in developing symptoms between the two arms. In essence, hydroxychloroquine wasn't effective in preventing people from developing COVID-19. In addition, there were more side effects in people who were on hydroxychloroquine arm. You might still find that Indian doctors are prescribing hydroxychloroquine. This study showed that 76% of doctors from India would consider hydroxychloroquine as prophylaxis. And this is because doctors in India have to follow local guidelines from ICMR and other bodies, whether it is rational or irrational, it doesn't matter. So they will probably have to continue to prescribe hydroxychloroquine as prophylaxis. Some of them, they take it themselves as well, as I know. Continuing on evidence about medications that don't work, there is something called lopinavir and ritonavir, which is a combination of antivirals which are used for HIV infection. This was a part of recovery trial and it doesn't work. Human monoclonal antibodies, these are medications which end in something called AB or MAB, something like infliximab. They don't work either. So what may work? There are works going on various medication, and I'll go through a very small list. I can't discuss everything. Azithromycin is an antibiotic, and on its own, we do not know whether it works or not. There is an azithromycin arm in recovery trial, and it's still ongoing. There is no result yet. In New York State, they sponsored a trial on azithromycin in March 2020. There was no news from them, and it might mean that azithromycin is not doing a wonderful job. Vitamin D is a speculative use. There are observations that low vitamin D may be associated with a higher incidence and severity of COVID-19. But another report eliminated the confounding factors, and they said that they didn't find any evidence of increased susceptibility in low vitamin D individuals. A trial may be done in the next 6 months in UK about vitamin D and COVID-19. Intravenous vitamin C has been or is being used. There are 6 ongoing trials, and there is no conclusion yet. There is no preliminary report yet either. Zinc has some role as cofactor in immune response, but I'm not aware of any trials that has an only zinc arm in it. There's something called interferon beta, and 3 doctors in UK found that this is useful in asthma, and they started a company as well. Interferon beta is part of Solidarity trial, which is run by WHO, and apparently it is showing promise. I don't know whether it is showing promise or not. It is definitely pushing the price of the share of this company that I mentioned up by quite a lot. There's something called ivermectin. It is an antiparasitic medication. And from Florida, there was a study called ICON study. And they reported in a preprint that they found a significantly lower mortality amongst hospitalized patients who had ivermectin. It has been very quiet since then in the last 6 weeks. So I'm not very hopeful that we'll hear good news about ivermectin very soon. And I had some personal communication from a friend of mine who did some research on COVID-19. He said that the part of the cytokine storm response is bradykinin mediated, and aspirin may have some role in that pathway. So aspirin in moderate dose, which is about 600 milligram a day, may have some role in COVID-19. We'll have to watch this space. Then moving on to vaccines, I'm very indebted to Dr. Pal for giving an excellent talk about the vaccines. And I'm not going to go through these slides in detail. I was going to say that you shouldn't put your hopes on a vaccine because it's not going to come in a hurry. And I was going to discuss the phases of vaccine development. The traditional timeline for a vaccine development is about 10 to 15 years. And what we are trying to do is we are trying to shorten that time span by overlapping various phases. So kind of academic research and preclinical trials are overlapped. Clinical trials in phase 1, 2, and 3, they are overlapped. People have said that they would like to start manufacturing early, but you can't really start manufacturing early unless you know at the end of phase 3 trial that this is something that's going to work. And then we can produce quickly, which is possible. And then if we go through the approval process quickly, and then the vaccine can be marketed. And realistically, I would say that I can't see the vaccine coming out in the market very soon. But we'll revisit this a bit later. Now, there are issues with antibody in the body after COVID-19, which, again, Professor Pal has covered very well. As I said, clinically, I had COVID-19. The swab test was not available for NHS workers at that time. When I was tested for antibody, I was positive. And few of my colleagues who had similar kind of symptoms were tested positive as well. And they went on to take part in a trial called SIREN, where every 2 weeks they have their antibodies checked and their swabs taken. And at least 2 of them have come back as antibody negative now. Generally, the notion is that the antibody stays in the blood for about 3 months and then disappears. Now, what does that mean? Does that mean that they are prone to COVID-19 again? Will they become reinfected? There are some reports that people have become reinfected. And if they are reinfected, will it be a less severe disease or a more severe disease? I do not know at this stage. I haven't read yet that antibody from the vaccine has disappeared. I haven't read anything like that in the things that I have read so far. And also, it raises the question that whether the vaccine need to be a multiple dose vaccine rather than a single dose one. There are reports that about at least 10 patients in the phase 1 trial of the Oxford vaccine required a booster. And then in the United Kingdom, we know that when it is going to be marketed, it is going to be reserved for essential workers like the doctors, nurses, teachers, and also for the people who are in the vulnerable group before it reaches general population. So these are the vaccines, and there are about 155 in research and development at this moment. If you want to keep track of what is happening to the vaccines, I would say that you use a reliable online tracker like this one from Guardian, the link of which I have given. And you'd find that there are 5 vaccines in phase 3 trial. It was 2 weeks ago when some of you might know that when I took part in another webinar. Of these 5 vaccines, 3 of them are Chinese, 1 is Australian, and of course we have the Oxford vaccine. There is Sinovac from China. They also have 2 vaccines from Sinopharm in Wuhan and in Beijing. There is this one from Australia. The Murdoch's Children's Research Institute is using BCG vaccine to boost the immune system. They are in phase 3 trial, and I'm very interested to find out what the outcome is. The Moderna vaccine is an mRNA vaccine, and there are a couple of things we need to remember about Moderna vaccine. One is that there is no mRNA vaccine that has ever been approved. And also, Moderna doesn't have any experience in producing vaccines before this one. So they are rather inexperienced in this line of the job. CanSino has already been mentioned by Professor Pal. And of course, Bharat Biotech, which had some input from Flugen, a company from America, and also from University of Wisconsin and Madison. It has moved from the preclinical stage onto the next stage. I was going to say why it is important to have antibodies and to have a killer T cell response, but this has been covered by Professor Pal, and going to say that the origin, because it produces both kinds of responses as we want a vaccine to do. So the next stage is that it needs appraisal of actual protection that it provides. Can it protect against severe disease? And if so, how long? The antibody from natural COVID-19 infection drops off. Will it happen with the vaccines as well? I don't know at this moment. There are a couple of or 3 points to note regarding this matter. I would say that this vaccine triggers immune response in a way different to natural infection. So the antibody from natural infection might drop off. The antibody from this one may not drop off. Then again, antibody may go down or may disappear from blood, but that doesn't mean that the body may not be able to produce an antibody next time there is an infection. There probably would be some B memory cells somewhere in the bone marrow stored to become activated when there is another infection or exposure. T cell response in case of other vaccines lasts longer than what we are noticing in Oxford vaccine, but this duration always varies, and I wouldn't too worry about that at this stage. Moving on, there is this other way of approaching the vaccine. Can one of the existing vaccines be used against COVID-19? I've already mentioned that in Australia they are trying to use BCG vaccine to boost the immune system. There are also some talks about the MMR vaccine. There was a paper in Molecular Biology within the last 6 weeks, which were asking that if MMR can provide some protection. And their argument was based on reports like there was no COVID-19 cases after measles campaign with MMR vaccine happened in American Samoa. Apparently, Hong Kong is another example where there was very good coverage with MMR vaccine. However, trials are needed, and about Hong Kong, I'm not too sure because they have gone through the initial wave and they have gone through second wave and they are in the third wave at this moment. In UK, I can say that most deaths are amongst older white people and also in Asian and black people. Most of them did not have MMR. Even I did not have MMR. I, however, had measles as a child, and blood tests showed that I had rubella as well. So my question is that, does natural infection give similar kind of protection? We do not know at this stage. I was not going to talk about Russian vaccine, but the situation has compelled me to do so. The Russian vaccine, we are getting various reports about Russian vaccine here and there. It's going to come tomorrow, it's going to come next week, it's going to come in the next 10 days, it's going to come in the next month. So this was in 30th of July in one local newspaper, and it was in Times as well. It's apparently it's available for public use by August 10th, but I looked further. Reuters said that the Russian COVID-19 vaccine is going to have a local regulatory approval, not a state approval. It's a local regulatory approval, and then they will have clinical trials. And the Russian Deputy Prime Minister is saying that Russia hopes to begin manufacturing the vaccine as early in September. And then I found last evening BBC is reporting that Russia plans mass vaccination campaign in October. It's not going to come in the next 10 days. It's not going to come on October 10th. And there was this interesting story about Newcastle vaccine, and they have come out with a preclinical report on 31st of July. And they are planning to manufacture a vaccine which would be like flu vaccine. I don't know the intricacies, that's not my specialty. But they are saying that if their method is going to be successful, they can use the existing mechanism that's there for production of flu vaccine to produce very cheap vaccine against coronavirus. And let's hope they are successful. But in the end, SARS and MERS, which have been mentioned by Professor Paul, they still have no commercial vaccine, and neither has HIV, which is an ongoing global pandemic, since it shouldn't give us any false sense of it and difference in approach, except a few countries which had something in common. The head of the states were women. India and UK, both countries were caught quite unprepared. There were poor test facilities in both countries, poor supply of PPE. The lockdown was probably late in the United Kingdom and probably early in India. Not enforcing strict test, track, and isolate and support in India was an error. In UK, the error was discharging elderly COVID-19 patients to old age homes, which caused SARS, killing up to 20,000, and someone is now saying about 30,000 or more older people. There were many mistakes. Some were similar in both countries, some were dissimilar. And we should learn from our mistakes and don't repeat them again. The difference is in United Kingdom, the NHS is free at the point of care and accessible to all. So even if you have no money, it doesn't matter. You'll be looked after. But in a money-driven health system, as in India, if you do not have money, you cannot get the best treatment that is available. And this will result in people dying. Also in the United Kingdom, the government was very visible. We could see the Prime Minister or his representative every day. They took questions and answered. And in UK, medical management was based on the available evidence. We got a directive or several directives every day, correcting the previous ones. Knowledge was shared promptly through WhatsApp groups and through Twitter. Doctors continued to work with or without adequate PPE. Management made quick decisions, and mostly correct ones in hindsight. And most people followed the lockdown rules. I wouldn't go too much into that because my comments might be interpreted differently with political uh, intention or not. So let's move on to what we can do, which are the do's and don'ts in COVID. So prevention is better than cure, but whose role it is everyone's role. And if you know what slows down the pandemic, do it. We do not need to reinvent wheel. And people who deny science or people dealing with serious science are a liability, and we should ignore these people. So social distancing is a must. Whether there is a lockdown going on or not, don't go out unless you have to. And if you go out, you maintain the social distancing. Wearing a mask is a must. Even a cloth mask is better than none. And there is no need for surgical 3-layer mask or N95 mask. I'll say why I'm saying this in a moment. I know of some people who are using the plastic face shield. Without mask, it doesn't give you any more protection than wearing glasses. Personally, I think gloves are good, but it is difficult to wear those in Indian weather. Some people are wearing gloves made of fabric and they are coming back home and washing it. That will do. Wear glasses, or if you do not have glasses, wear sunglasses. That gives some protection to your eyes. I can't show it to you because I'm not in picture at this moment. When I'm at work, I'm using a special glasses, which doesn't have the power, but it protects my eyes so that I can't really touch my eyes with my finger. So in my opinion, masks, gloves, and glasses are to stop giving your germs to others and to prevent you from touching your eyes and your mouth with your fingers. Your mask protects me, my mask protects you. Don't get too technical with this. We are not going to perform surgery. We're just going to prevent spread of the infection. I would advise using a head cover because droplets can land and rest on hair and come back home. Any fabric cover will do the job, and when you come back home, just wash it. I would suggest avoiding plastic ones. And wash. Every time you come home, discard all clothes, take a shower with soap or a body wash. Wash your glasses or sunglasses. And by the way, you don't need Dettol to wash your clothes. Simple soap water will do. If you are using your mobile phone outside and if it is water resistant, then come back and wash with soap water. If it is not water resistant, use sanitizer to wipe it clean. If you are using coins outside, you wash them with soap water. About paper money, well, I wouldn't say that you use soap water or sanitizer for your paper money. I probably would leave it in the sun. Apparently, ultraviolet ray kills everything, but I don't think that we know how long it takes to kill the coronavirus. I don't think anyone knows. Apparently, ultraviolet C sterilizers are being made and will be available very soon, but I think they would cost more than 10,000 Indian rupees, if not a lot more. Keep your shoes separated. You can wipe shoes with antibacterial wipe, but nothing else will be needed. Plastic packets from shops, I would say wipe it with soap water and let it dry. And follow lockdown rules. There is a paper in PMG in the last fortnight which shows that closure of schools, workplaces, and public transport, restriction on movement and mass gathering, and public events slow down the transmission of the virus and flatten the curve. Paradoxically, though we are advised not to go out, we are more likely to catch the virus in poorly ventilated spaces, often in offices with air conditioning which goes through ducts and connects various rooms together. So therefore, work from home if you can. And at home, open the windows which face the sky to increase ventilation. You can run the air conditioning at home as long as it is in a single room. Pressure is much better. And when you are recirculating the air in the air conditioner, just think that if any of those people who are in that room has asymptomatic COVID infection, it's just going to give the infection to everyone in the room. Now, it is airborne, or are there questions about airborne? I think there was a question about whether it is being airborne in the that. Don't worry about it. WHO says that they can't rule out the possibility that it could be airborne, but they didn't say that it is airborne. And they warn about aerosol generating procedures, which are usually happening in the hospital or when people are ventilated. It can also happen in the dentist's chamber when they're doing the dental work, and also in some indoor crowded places like classes, but not at your home. And as far as and put it through a PC that is proven, unless that paper comes out, I wouldn't worry too much about the virus being airborne. And yes, your freeze and freezer is not full of COVID-19 either. There are reports of outbreaks of COVID-19 in meat industry, but you need to remember these people, they work in rooms, the big rooms, which are kept in freezing temperature. Temperature around 4 degrees centigrade can be a sweet spot for COVID virus because droplets can freeze and can spread to co-workers. It can travel more than 2 meters in that kind of situation. We at home, we do not work inside our freezer, so there is no need to panic. Use your fridge and freezer as you have been doing all these years. There is no need to change. If you are buying fruits and vegetables, wash it in running clear water or keep whatever you want. No one knows how long you should be doing so. So 10, 20, 30, whatever you want, you can do. Do not use your sanitizers on these. Do not use soap water on these. And you don't need warm water for these either. Fish or meat, as far as I remember, or as far as it is done in our house, whenever fish or meat is brought to our home, we wash it with water. Then either we proceed to cook or we store in the freezer and freezer. Do the same. And always remember, the cooking will kill the virus. And no, a clove of garlic is not going to protect you from COVID-19. There may be beneficial effects, but it's not going to protect you from COVID-19. Whoever advises this, always ask which trial has been conducted to prove this. We should not give false hope to people. A lot of people buy over-the-counter medications for boosting their immunity. Let me tell you, it doesn't boost your immunity. There is no over-the-counter medicine, there is no vitamin, there is no diet that boosts the immune system. Besides, many people die from COVID-19 due to cytokine storm syndrome, which is an over-responsive immune system. So you don't need to specifically boost your immune system. And we had a very nice chat about herd immunity from Professor Paul, and I'm glad that he also stressed on the point that there cannot be any herd immunity without a vaccine. And if you are living in a sky rise, then, and if you have someone in your, well, even if you do not have someone in your sky rise with COVID-19, you always clean and sanitize lifts and stairwells. Only yesterday, I had news from one of my schoolmates whose when someone in the accommodation, not in his flat, in someone in other floor, had been diagnosed with COVID-19. So people came and sanitized that person's room or flat, but they didn't do the stairwells, and that's where you are going to catch the virus if you are going to catch it. So if your neighbor has COVID-19, please do not panic. Use normal precautions and use mask, hand sanitizer, and wash hands more often, up to the elbow at least. Hard people panicking and agitating is not going to help anyone. And when you are using a hand sanitizer, just make sure that the alcohol content is more than 60%. Otherwise, it's not going to be effective. So let's come to the worst situation. Suppose you have COVID-19. Can you be managed at home? I would say that you should follow what your government says and what your doctor says, and don't follow what I say, because situations are not the same. In the United Kingdom, COVID-19 cases were managed mostly at home. Only in case of respiratory symptoms, they were admitted to the hospital and managed there. So COVID-19 can be managed at home. It is possible, but it can be risky as well. The first thing first is that you should know the symptoms of COVID and ignore what ICMR has said about fever being a symptom of COVID only in 17% cases. They studied people who were hospitalized. They didn't study people who were in the community. If you're in the community, fever and cough remain one of the major symptoms, whether the fever is low grade or high grade. Usually fever is low grade, less than 100 degrees centigrade, 100 degree Fahrenheit, or nearabouts. And I've given a very nice link which links to the ever-changing symptoms or list or growing list of symptoms from COVID-19. And remember, the ACE2 receptors are in other places of the body as well. So abdominal pain and loose motion can be a symptom of COVID-19 as well. But the good news is that 80% of affected people have mild symptoms. 15% may need oxygen, and only 5% need ITU. And it is only those people who need ITU may die. You should also think about comorbidities. Any long-term medical condition makes a person vulnerable. So diabetes, chronic kidney or heart or respiratory disease, etc., will make someone vulnerable to COVID. And do not assume you have immunity because you are taking something. No one has immunity. At home, you should isolate yourself in a room with attached bath. I know it is very difficult in houses or flats in Kolkata. If there is no attached bath, you should arrange for something what we used to call as sponge bath. If you are using the commode, and after every bowel movement, you should close the lid before flushing the toilet. You should wear a mask when you are coming in contact with others in the house, and they should wear a mask too. If you have sore throat, you should gargle or you should do some steam inhalation. Wash your hands regularly and avoid touching face, mouth, nose, or eyes as much as possible. You should write down your symptoms as you are in isolation because no one else can monitor you. You should keep yourself well hydrated and well nourished and enjoy the isolation. Read, write, listen to music, make video calls, and avoid WhatsApp University. Take regular paracetamols, 650 milligram 4 hourly, and that will help with myalgia as well. If you have a pulse oximeter, and you can use one. If you have never smoked, any reading above 92%, well, 92% and above is all right. If you are a smoker or an ex-smoker, it should be 88% and above. If it goes below that, then yes, you should panic. If you don't have a pulse oximeter, then you can do a very small test. Take a deep breath in, fill your lungs with as much air as possible, and then count as you breathe out, like 1001, 1002, 1003. The idea is to achieve a counting speed of a number per second. And if you practice now and do it now, that will give you your baseline. and you are free of disease at this moment. Now, if you develop any respiratory symptoms, if you count in similar way, then it tells you if your breathing is affected or not. There is no parameter that if you count less than 10 less than what you normally do, then you are affected. There is nothing like that, but it gives you an idea about your breathing and how it is going. Some doctors, some corporate hospitals, and there is also something called a COVID care network which is being developed. They are going to run or already running some home management schemes. You can get enrolled with them. You should take their advice, and their parameters will be different from what I have said. You should follow them, not me. Even when you start feeling better, like you have no fever, your cough has improved, you should remain in isolation for up to 14 days, or whatever your doctor or your government recommends. I wouldn't advise that you should buy oxygen cylinders or keep oxygen cylinder at home. If it is used constantly, it will last for about a few hours. And if you need oxygen at home, you should be thinking about hospital. So if you are struggling to breathe, or if you have a chest pain or pressure-like feeling in your chest, if you have increase in temperature, or if you have persistent temperature with no remission despite maximum dose of paracetamol, you should think about going to the hospital. Delirium, especially in elderly, like hallucinations or inability to think clearly, Bangla jeta amra boli bhul boka, that is also a sign of COVID. Apart from that, there can be severe weakness, muscle and joint pain. All of these might need hospitalization. At this stage, you should remember at least two things. There is something called happy hypoxia in COVID-19 pandemic. We have seen people with COVID-19 and they have lung changes, but they are tolerating lower oxygen in their blood and suffering from no respiratory distress at all at rest. The explanation is unknown at this moment. I would also say that you should be lying in prone position if you have any breathing distress. Prone position means that you should be lying on your belly. That improves oxygenation and pulse oximetry readings. Next, we will move on to preparation for winter. The winter is fast approaching, and naturally people are worried. Well, I don't think it's fast approaching, but we are making preparation for the winter. In UK, we are informed that with influenza and the Brexit, COVID-19 is going to hit us hard during the winter. The wave can be 3 times the initial wave that we have endured. However, there are different analyses which says that COVID-19 is not that affected by seasonality. So the jury is still out, and it's the policymakers, they will decide what we are going to do. But what we as individuals can do in preparation for the winter. With the COVID-19 pandemic, our options are limited. Some of the established methods that we use to stop an endemic are unavailable. So we can't stop the origin of the virus because it has spread worldwide. There is no effective treatment for the virus. We can't improve herd immunity since there is no vaccine. So the only way out is the fourth, which is breaking the transmission chain. Therefore, the current advices on measures to break the chain of transmission are here to stay. So all current advices are still going to be valid in winter time. Then we should risk stratify ourselves. I would say that you should know your blood group. It is not formally established, but a preliminary report indicated that people with blood groups B, O, and AB are less susceptible in comparison to blood group A. The link is there. You should also know the dynamics of the disease, and it is going to stay with us for some time. Few months, if not few years. I hope it's not few years. Another preliminary report says that people are most infectious in the first 5 days of the infection. Though people continue to shed virus after that, it doesn't mean that they are infectious. and nobody so far has proven to be infectious beyond day 9. So that means that if we go into self-isolation, it used to be 7 days in the United Kingdom. Now we should go for self-isolation for at least 10 days if we develop symptoms suggestive of COVID-19. The other thing is that you should know your symptoms. Is this your usual winter cold or is it different? In that list of symptoms of COVID, you'll find that runny nose is not a symptom of COVID-19. There's no harm in taking vitamin D or vitamin C supplements. You can use in winter the oranges, the limes, the lemons, the chili, and amla. If you're taking a multivitamin tablet, I would say that usually a balanced diet contains everything. And water-soluble vitamins are not stored in the body. So in my opinion, tonic or supplemental tablets are not necessary. Vitamin supplements in cooking oil are likely to be rubbish. When you cook, wouldn't the heat denature the vitamins? If you want to take garlic or ginger or coronil of Baba Ramdev, it is fine. I have nothing against it, but none of these are going to boost your immunity or save you from COVID-19. Don't reside in a bubble of false security. And look after yourselves. One of the problems with the lockdown is that we are putting on weight. We need to reduce weight. Excess fat can affect the respiratory symptoms or system, and it is likely to affect inflammatory and immune function of our body. Aim for a BMI which is less than 35. An ideal BMI is between 18.5 to 24.9. To reduce weight, you should exercise. Even walking within your flat is good. But you should also reduce food intake. The total calories should be low. If you develop cough and cold, cover your mouth with a tissue or cough into your elbow and then dispose the tissue off in a separate bin and wash your hands immediately. If you have sore throat, you can gargle or use steam inhalation. Dettol or Betadine in water for gargling does not add into anything. It's not needed at all. Lukewarm salt water will do. A dispersible aspirin, well dissolved, might help in reducing soreness. You should take paracetamol to keep your fever down. You should stay home and self-isolate even with minor symptoms such as cough, runny nose, mild fever until you recover. And as I said, 10 days at least. Use only trusted sources. Despite the negative publicity, WHO is quite reliable. I'll speak a little bit about that later. And finally, don't panic. In 4 out of 5 who are positive with COVID-19, it is a mild condition. And use your helpline. And yes, avoid WhatsApp University. We're already confused with studies and their conflicting results. And even we as professionals, we find this is very worrying and it's very difficult to make up our mind. So for common people, it's even more difficult. So before forwarding any news item, please check if it is authentic or not. If you can't check, don't forward. WHO has a very good Mythbusters site. And it will tell you about various things like whether shoes can spread COVID-19, whether drinking alcohol gives you any protection against COVID-19. It doesn't. Exposing yourself to sun or temperature higher than 25 degrees centigrade, it doesn't give you protection from COVID-19. When first COVID-19 broke, people were saying that, oh, it's not going to survive in Indian summer with temperature of 55 degrees Celsius. It definitely has survived the Indian summer. Rinse your nose with, if you're rinsing your nose with saline, it does not prevent COVID-19. Don't do it. And also beware of these memes. I'm sure this gentleman, Mr. Seagal, is trying to make fun of the scanning device. But some people might believe in this. And would stop people from scanning themselves. And yes, Russian vaccine again. Now, can you tell me, when even Russia says that they are going through various stages and it is not final yet, how the noetic buzz in Facebook can say that it has gone through and passed every trial? And are you going to believe the noetic buzz anymore than the news agencies like BBC or Reuters? And of course, there are things like this when someone from Bangalore says that he has US FDA and European Union approval for this medical device that can neutralize coronavirus. And he says that it is going to emit electrons and electrons are going to kill coronavirus. Now, I said that it's probably false, it's probably fake. I was told that no, it was in a 24-hour news channel, it can't be a fake. So I thought that maybe I should have a look into the US Food and Drug Administration website and look for the things that are mentioned, like Shikocan about this device, or corona neutralizer, or scaling hypercharge corona cannon. And there is no result for either of these. So I'm not sure. Shall I believe in FDA's website or shall I believe this news item? I don't know. And also, there are these kind of things in the internet. So this is an advertisement came in the last couple of days, and it shows that this goat at least has teeth in where it shouldn't have. So all these things are there. So don't believe anything that's going on in the internet. So aim for balance in your life. Think about physical balance, do exercise. Think about spiritual balance. So stop and take a look at where you're going with your life. Think about mental balance and learn something new. Teach something new. Stay engaged. Keep yourself busy. And think about social relationships. Look after your family. Friends. Talk to each other more. And browse less. Try to develop some. So in conclusion, I'm not sure how much ray of hope I was able to give, but be assured, all over the world, people are trying to find a way out of this pandemic, and we shall overcome. The COVID-19 has shaken the self-belief in all of us, the human beings. But this is the time to stand firm and deliver in whatever small capacity that we individuals have. I'm going to end with words from Kobi Bipode Amnachana Kobi. Thank you.
Thank you, sir, for your very informative and outstanding presentation. We hope your lucid presentation may rule out the panic and doubts about COVID-19 from our minds in this pandemic situation. Now the session is open for question and answer discussion. There are some questions in the chat box. Please see the chat box. I will read out. Number question 1. Sir, current. Yes.
Yeah, so shall I start with this one? What is the point of origin of this virus?
Yes, sir.
Yeah, I don't know. I have no reason to disbelieve what the Chinese authority has said, that it kind of originated somewhere in a market in Wuhan. There are lots of other people who are saying that it can be developed by Americans or it has been developed by the Chinese themselves. I'm not someone who believes in conspiracy theory unless I have any different evidence. I think it is what the Chinese authority have said. Besides, I'm not a virologist, so it's very difficult for me to answer that question. So the second question is that can flu vaccine reduce the severity of COVID-19? Answer is no. So far, no existing vaccine has been proven to be of any benefit against COVID-19. The candidates are the BCG vaccine and also this MMR vaccine, but the research is ongoing, result is not out. So are there any cross-reactivity between the antibodies present in heterogeneous mixture of a donor's plasma with the receiver's own immune cells? That's a bit difficult for me. I'm a geriatrician. I'm not someone who deals with all this immunological stuff. But from my knowledge, what I can say is that it is possible that the donor's plasma might have some kind of antibodies which might work against the receiver's own immune cells. But it is very unlikely that's going to happen. And as I have said, that the preliminary report from USA said that the plasma, the treatment with plasma seems to be very safe so far, and there doesn't seem to be any significant adverse effect. So I wouldn't worry too much. The other fact is that you are thinking about using plasma for treatment is in patients who are quite advanced as far as the condition is concerned, people who are probably already on ventilators and are not doing very well. Why diabetic people are at high risk of COVID-19? Very good question. The exact answer is not known, but the theory is that since diabetes is a multi-system disorder and it has more effect on various organs or organ systems of the body than it shows. It's just not a rise in blood glucose, it affects everything. And it makes the body in general weaker than it should be. And that's why probably it is more prone. As I said, we don't know all the answers yet. The next question is that is the BCG vaccine going to provide us better immunity, or is it is just a myth? I wouldn't say it's a myth. There is definitely there is some let me rephrase it. I wouldn't say it is a myth. There is definitely some merit in the hypothesis, and that's why people in Australia, they have taken this up and they are doing a trial on that, which has gone into phase 3 trial, which is a good thing because they have passed through the phase 1 and phase 2, and there is probably some merit in continuing the trial. So who knows? Probably because in India we had a very good coverage of BCG vaccine at certain in certain years, not throughout, because I didn't have BCG vaccine. My wife had. No, she didn't have either. My daughter didn't have it because she was born here. My son had it. So it's not very consistent, but definitely the coverage in India with BCG vaccine is much more than in other countries. And maybe that is one of the reasons why the actual death rate, if we consider it, the number of deaths per 1,000 people or per million people, is lower in India in comparison with other countries. So it's not a myth. There is some substance in it, and it's being investigated. We'll know the result in a few months, maybe. Can ACE inhibitors inhibit ACE2 receptor? Well, yes, that's what they do, but they do it in the heart. So the general advice was that people who are on ACE2 inhibitors, they don't need to stop the ACE2 inhibitors because they might be having or suffering from COVID-19. So there is absolutely no reason to stop the AC inhibitors. If someone is taking it for cardiac cause, it is much better to continue to take it. The other thing I would say is that one of the features of COVID-19 is that it can cause myositis, it can cause viral myocarditis. And there are reports of people coming to the emergency department, especially in USA, who came with features of a heart attack with changes in their ECG, but they went on to have angiogram, and the angiogram is normal, and they have been found to be COVID positive. So don't stop this. It's going to cause more problems. Right? Any other questions?
We don't have any other questions.
Sorry?
Yes. Yes, I think we don't have any other questions. Then
Just a moment. YouTube, someone has how effective humoral immune system can prevent a respiratory tract virus like COVID-19, as no blood cells is known to enter the respiratory tract or lungs. No, the blood cells are in the respiratory tract or in the lungs. In the walls of the alveoli, on one side you have air, that's on this side, and this is the alveoli through which the capillaries are there, and capillaries contain the blood cells, and of course the red blood cells, which take oxygen from air and release the carbon dioxide. Now, what happens? I don't have this picture here at this moment. It wasn't in the presentation, to be honest. What happens with COVID-19 infection is that the membrane that separates the blood from the air, that becomes damaged or becomes what we call leaky, and it will leak serous fluid inside the alveoli, blocking it. And if it happens to several alveoli, then it will go give rise to consolidation of those air-filled balloons, and this is called pneumonia. So normally, yes, you are right. Blood cells and the bloodstream is separated from air, but it's there in the lungs. And if you think that the cells, the tissues in the lungs, they will have to get their nourishment from somewhere, and they are not able to get it from air. So there is some blood supply to all these respiratory cells. So it's not quite true that no blood cells are known to enter the respiratory tract or the lungs. There is some blood supply for the tissue, and there is also some blood supply which takes part in the exchange of oxygen and the carbon dioxide. I think that's all, isn't it? Yeah.
thank you again, sir, for your valuable time and a very nice presentation. Now I am going to transfer the session to Dr. Priya K. Gopal. It's over to you. Thank you, sir.
Hello, am I audible?
Yes, you are audible.
So, thank you so much, sir. And after these two wonderful and very informative lectures, we have come to the concluding part of our webinar today. And may I now request Mrs. Ambalika Chakraborty of the Department of Chemistry, Muralidhar Girls College, to deliver the vote of thanks. Ambalika Di, over to you.
Thank you, Priya. Am I audible? Thank you, Priya.
Yes, you are audible.
Am I audible?
Yes, yes.
A very good evening to all of you. It's an honor for me to deliver the vote of thanks at the end of today's international webinar on behalf of our esteemed institution. First of all, I would like to thank the distinguished speakers, Dr. Shantanu Paul and Dr. Somadipto Bandyopadhyay, who have spared their precious time and enlightened us with their enriched and very informative speech. I would like to extend my sincere thanks to the patron of today's webinar, our respected principal, Dr. Kinjolkini Bishwas, for taking the initiative and guiding and inspiring us in conducting today's program. I would like to express my heartfelt thanks to all the participants for overwhelming response and their enthusiastic and active participation. I would like to thank the ICT cell of our college for the technical support to make today's event successful. Again, thank you all and stay safe.
Priya, do we end the program?
Yes, we are done with the program, Didi. The session is over.
Principal ma'am, do you want to say anything? Please unmute your mic, ma'am. There is again some problem with ma'am's mic, I think. Okay, then we are ending this today's program.
Yeah, okay.
Okay.
Okay, ma'am.
এতক্ষণে হলো
Just a last few things. Those who have yet not filled up their feedback form, please fill up the feedback form, and the e-certificates will be delivered to you directly to your registered email address. Ma'am, you have unmuted now.
at last.
Yes, the mic was disturbing you very much today.
Yes.
Yes.
Okay then. Thank you
Okay.
very much.
We had a wonderful presentation today.
Insight into the
Yes.
COVID. Okay. Bye.
Bye.